So the big question is how does bacteria survive? If you think about it, if the bacteria cell is going from a lysogenic state where, yeah, it's making more and more cells, but then it has viral DNA in it, no big deal. But then it changes into a lytic cycle, and that bacteria lyses or explodes. Why do all bacteria not essentially lyse and become extinct? So there's three solutions or three answers that bacteria have in terms of how they defend themselves against a phage. So the first one is just natural selection. So over time, when these viruses are infecting these bacteria cells, over time, only some of them are going to survive. And the ones that survive are the ones that have a different surface. So please remember that the viruses attach to the outside of a cell. Viruses can only attack certain types of cell with like a locking key on the outside. So if for whatever reason there's a bacteria cell that has a different membrane on the outside, in that case the virus isn't going to attach and attack that cell. And that cell that survives can then replicate and replicate and replicate free of harm of that virus that was originally trying to target it. Another type of bacterial defense is the use of restriction enzymes. So restriction enzymes restrict the phage's ability to replicate inside the cell should it be infected. It does so by destroying the viral foreign DNA. And in that case, it's broken down. Also, the DNA that's inside the cell itself, the good bacterial DNA, it is methylated, which will prevent it from being attacked by that virus as well, and trying to make it be degraded, essentially. The third type of defense against phages and viruses is what is known as CRISPR. So CRISPR is a new method. Okay, so picking up where I left off, the third type of bacterial defense is what is known as the CRISPR case system. So without getting into too much detail, when viral DNA gets inserted into that host genome, often it's in between repeats of DNA. So if you look at the picture on the right, you can see the brown genes, and then there's a blue and a green and a yellow. Those are going to be the different colors. The bright colors are going to be the viral DNA that has been inserted via being lysogenic. Okay, so the cell is making more and more cells. That viral DNA is within the genome. The cell is not lysing. So in this process, transcription occurs, so RNA is going to be synthesized, and there is another protein called the case protein or case enzyme. So this is going to look for RNA that is essentially being repeated. When it binds to that RNA molecule, the repeated parts get cut out. The repeated parts, along with that viral DNA, get cut out. And then it gets spliced back together. It gets spliced, and then it gets basically put back together. And then the viral phage DNA is gone, along with those repeats, and the cell is free of that viral DNA in this cell and in future cells when it replicates. So there are a few important things about animal viruses and how they affect animal host cells. One of the key components is what type of genetic information does that virus have? Is it RNA? Is it DNA? Is it single or double-stranded? The other thing, and I'll get more into that in just a moment, the other thing that's important is there an envelope on the outside of that virus? So if there is an envelope, the host membrane is going to almost invite the virus inside, because the host membrane is going to recognize that envelope, almost like it's recognizing itself, but instead it's a virus, because that envelope is made of glycoproteins. And on the outside of an animal cell, often it can recognize these glycoproteins as good things, which in fact a virus is not going to be a good thing for the host cell. So the host is essentially tricked, and the virus is able to get inside of that cell. And then it's going to be difficult for that host to detect the virus, because in this case, if you look at the diagram, the RNA that's inserted into the cell is going to undergo translation and build a protein, and the ER is going to make more glycoproteins, and all the stuff gets put together, and all of a sudden we have a new virus which is going to exit the cell via exocytosis. Please remember that different types of viruses can have different types of genetic information. It can have RNA, DNA, and of that, it can be single or double-stranded. So of the different types of viruses that affect animals, there's 3 types of single-stranded viral RNA, again, genetic information in these viruses. So one type, what it does is it serves as actual mRNA. So once that viral RNA is inserted into the host cell, translation can automatically occur. And by translation, meaning a polypeptide is going to be built, a viral polypeptide. It's going to be beneficial for that virus. A second type of single-stranded RNA molecule, again being the virus's genetic information that's going to get inserted into that host cell, operates like a template for mRNA to be built. So in this case, it's almost like an imposter, like a DNA imposter, but off of the single strand of RNA, a new version of mRNA is going to be transcribed or transcripted. And then normal translation will occur, and a virus polypeptide, again, some sort of a protein that's going to benefit that virus, will be synthesized. The third type is known as a retrovirus. So this is going to, again, this is a single strand of RNA that is going to go into that host cell and use reverse transcriptase. So from this virus's RNA, it's going to operate again like a template, but in this case, DNA is actually going to be synthesized. From this newly made viral DNA is going to be inserted into the actual chromosome of the host. So in the normal DNA, you are going to have, again, bits of viral DNA, which I had already mentioned in the past lecture, which is known as a provirus. So now the host transcribes or does transcription with that provirus to make new virus parts, again via translation and building that polypeptide. So an example of a retrovirus is HIV. I'm going to go into a little bit more detail on how it affects the cell on the next slide. Okay, without getting into too much detail about how a retrovirus and specifically HIV are able to infect cells, I think the picture summarizes, again, as often with these chapters, pictures can really help with the visual. On the upper left is going to be the HIV virus, which again is a retrovirus. It is able to infect the cell by having that glycoprotein exterior, which again almost invites the virus into the cell via the signals on the outside of the host cell. So once the virus is inside, it is able to release its viral single-stranded RNA molecule, and reverse transcriptase is the type of enzyme that comes along with that viral RNA molecule, which that is the enzyme that is going to synthesize viral DNA. And once there's viral DNA, it is entering into the host nucleus. It makes itself a part of the host genome. So if you look in the middle of the diagram, you see the purple circle, that's the nucleus. And then the viral DNA, now known as a provirus, has inserted itself into that chromosome. And from there it is able to make other RNA molecules, viral RNA molecules, which get synthesized within the cell, thanks again to the different parts, but the ER helping to build glycoproteins for the outside. And then all of a sudden we have a totally new virus that is able to exit via exocytosis and look for a new host cell to infect.